professor of ecosystem biogeochemistry in the Department of Land Resources and Environmental Science here. He's been here since 2009. Before that, he did a postdoc at Princeton, and then he got his PhD at Virginia Polytech and his master's at Oregon State. Yay, Oregon State. He works on, he looks at ecosystem dynamics from a biogeochemical perspective. So I think for that it means how do ecosystems respond to things like climate change, disturbance, and people, which is near and dear to many of our hearts. Jack's also co-director of the Environmental Analysis Lab that's in LRES, and he's editor of the Journal of Biogeochemistry. So turn it over to you. Thanks, Jack. Thanks for coming.
Thank you. Thank you all for coming, and thanks for inviting me again to speak at IOE Rough Cut. It's a real pleasure to share what I've been thinking about over the last couple of years. And so to cut to the chase, to the title, I took this definition of causality straight from Wikipedia, and it's a fine one. It is the relation between an event, the cause, and a second event, the effect, where the second event is understood as a physical consequence of the first. It's limited because it doesn't quite capture feedbacks that are inherent in complex adaptive systems. And the debate about what is causality has been occurring at least since the time of Aristotle, probably captured best and formalized by Berkeley in the 1700s. And basically, his formalization of what causality is and cause and effect hasn't changed very much. Analyzing it quantitatively, probably the best contribution over the last several decades derived from economics was Granger Causality. And then I'm going to be discussing a method that I've used called Convergent cross mapping to try to understand causality in complex systems. Okay, complexity, I borrowed this from Simon Levin. And this is an interconnected network of components that cannot be described by a few rules, generally manifest in structure, order and functioning, and emerging, this is an important concept, from the interactions among diverse parts. So I'm going to emphasize these two themes, these two ways of knowing, to address two major problems, and that is how ecosystems are responding to present day and past climate change, and how ecosystems respond to disturbance, and how we model that, and how we scale the response of disturbance from individual organisms to the ecosystem scale. So the two broad questions that I have that I'm going to address using case studies in Montana ecosystems today. How have changes to background aridity affected terrestrial ecosystem dynamics, and how does dryness interact with other vectors of change, other vectors being things like CO2 increase, CO2 fertilization effect, nitrogen deposition, direct effects of warming. And the second chapter of the talk really is how do ecosystem level patterns of nutrient limitation and primary production emerge from local organism specific interactions as ecosystems get older and as the component organisms get older as well. And as organisms get older, they tend to get bigger up to a certain point. And there's a lot we don't understand about how that works, especially after a disturbance. Okay. The two systems that I'm going to be discussing today are the Swan Valley, beautiful area in northwest Montana, and the Bangtail Mountains, just 30 kilometers northeast of here. Okay. For the first part, I'm going to be addressing subalpine grasslands in the Bangtail Mountains. And there's been a lot of interest almost every day on NPR and New York Times. You see a story about drought, increasing drought severity, predictions for increasing aridity, and especially in areas that are already warm and dry. And there's just been a flurry of global change experiment papers in the high-level journals like Nature, Science, PNAS over the last several years, doing very controlled randomized experimentation with different global change factors. For example, increasing CO2 concentration and watching how plants grow, adding nitrogen, warming a system. And these are mostly conducted in grasslands. Why? Because grasslands respond faster than other ecosystems because they don't have woody vegetation. So things can be quicker. You can see a response within a few years. These are very powerful, strong inference studies that are really important for understanding the mechanisms of how ecosystems are responding to these global change factors. But they're limited in a couple of ways, as important as they are. One, they don't capture the long-term dynamics of how ecosystems really respond to ambient changes in temperature, CO2, and other forcing factors. Transitions, tipping points, oscillations in climate and weather. And all the authors of these would acknowledge that the rate of change is obviously unrealistic, especially within a given funding cycle. CO2 doesn't double in 5 years, and nor does temperature. But this is what they do, and they see strong effects, and they get published in Nature. Okay, so what they can't address, really, is long-term background changes to things like aridity. Now, this is not drought. Drought is a discrete major disruption, a change in dryness over a period of time. But there are a number of modeling studies suggesting that background aridity, this is the dryness of terrestrial areas, is increasing globally. Of course, not everywhere, and it's particularly strong in warm areas. Surface humidity over the oceans is generally increasing. And just because evaporative demand can't keep pace with changes in vapor concentration, many areas on land are getting dry. This is something very hard to capture in a short-term experiment, of course, and we really don't know how these sort of changes interact with other background changes to, like, for example, the nitrogen cycle, in which we have more than doubled bioavailable nitrogen inputs to the terrestrial biosphere, and warming and CO2 concentration increases that we are all familiar with. So how do we address this? We have a number of ways. One, we have satellite systems that go back at least 20 years, and these are very powerful large-scale sensing of drought effects using things like NDVI. But there is also other opportunities, and these are long-term data sets that go back to the 1960s. And one of the most important of these is the International Biological Program. Okay, some of you guys are probably familiar with this, but this was a huge international effort to organize scientists from around the world to understand really fundamental questions like, how do biomes work? And why are biomes where they are? And the positivism and enthusiasm captured in this is so great. These models, as they develop, will not only provide understanding, but also when we build a highway, dam, city, or pipeline, predict the consequences. And are we there? I'm not sure. So one of the largest of the IBP studies was the Grassland Studies, and arguably one of the most successful as well. These are the sites established in the 60s, and there's one in our backyard. This was called the Bridger Mountain Grassland Site. It's actually in the Bangtail Mountains, but apparently Sims found the name inappropriate. So, and this study has also been continued by our own Tad Weaver since 1968. So he was an original cast member, and he has been collecting harvest data as an approximation of net primary production in these grasslands almost annually for 45 years. It is a remarkable, globally unique data set that we're lucky to have access to. Okay. So this is the site. It is also home to the world's longest continuously running snow manipulation experiment, as far as we know. So it's an Idaho fescue grassland, subalpine, 7,500 feet high. We have control areas over here. This is an area in spring. You can see the snow fences that effectively create 4 times and then 2 times ambient snow depth. And the system gets very dry in the summertime. It is windswept. It's a pretty harsh environment. So the peak growing season is May and June. And it's a lot of snow and it's cold. So a very short growing season. Okay, first time revealed to the public. What has happened over the last half century to primary production in this grassland? We've had possible changes in nitrogen deposition. CO2 has been rising. That should make plants grow faster, right? But it's a dry system too. We have observed a 50% decline in net primary production over the course of this study period. This in gray is total above ground biomass, is approximation of above ground net primary production. These are grasses, these are forbs. The most severe decline is observed in forbs and wildflowers, and the data bears that out, and anecdotally TAD says that the number of wildflowers have decreased dramatically over time in this system. And you can see all sorts of complex dynamics. This dip right here corresponds to a major drought in the late 80s. There's another drought here. We do see something, but then it bounces back. Forbs never really recovered. And in the inset here, in the gray, is basically the cumulative reduction in above ground carbon uptake. And so basically the capacity for the system to take up carbon has been cut in half over the last half century. Why? Getting to the cause. What generated this pattern? We first looked at climate patterns. These are regional from our own weather station here and the Bozeman Airport. There's been increases. Doesn't look like it much here because of the scale, and temperatures like most places on Earth. At least around here in the valley, there hasn't been directional change in snow or snowpack, and this is borne out by local snow tull sites as well. This is the Palmer Drought Severity Index, a commonly used derivative index of overall dryness of the system. But the most pronounced regional change, as far as I know, has never been reported till now, is a 50% reduction in late summer rain. This is true here. This is true at BZN Airport. This is profound. And I'll get to the implications for that later. Again, I said most growth is occurring in the early spring, so why would that matter? Well, it is interesting. So again, just showing there's regional coherence here between MSU and BZN and in terms of August and the late summer precipitation. It's declining, although if you're around here this last August, you might be skeptical. The overall pattern is going down. So there does seem to be a visual, intuitively appealing correspondence between the proportional slopes and declines here and the decline in above ground biomass. Problem solved. Let's just regress these things against each other. And it's very tempting to want to correlate. And you see this quite commonly in the literature. It's very scary to do so. If you actually run the correlations year for year, the correlation is terrible between any of these climate variables and above ground production. You know, for those of you guys who work on time series, this shouldn't be surprising because it doesn't incorporate complex dynamics, lag effects, and other things that are inherent to complex systems, and especially when you're looking at systems over the course of time, how systems evolve. So we all know from Statistics 101 basically that correlation is not causation, but moreover, correlation is not required for causation whatsoever. You can have in dynamically coupled systems things that are very causally coupled and they show no correlation whatsoever. Here's an example of this. This is a two-species dynamic difference system with nonlinear with chaotic dynamics, and this is one run of the simulation, and you can see at one period, one window of time, what appears to be strongly correlated dynamics between two variables. We can call this snowshoe hares and lynx if you want. And then an area where there's absolutely no correlation or anti-correlation. scenario where it's partly correlated, but maybe a lot of time series look like this. This is very common in nature. Many causally coupled systems are weakly coupled. And so what do we do to address this? Again, I said one powerful way is Granger Causality. And what that basically says is, out of the whole universe of possible contributing factors to an event, if something is said to be Granger caused, if we remove all other of those factors and it's still driving the pattern. Okay, well, this works only in a purely stochastic world, and an alternative approach, which derives from dynamic systems theory, is convergent cross-mapping. And here I lean heavily on, and I'm thankful to the insights of George Sugihara here at Scripps. And so he and Robert May have been working on this problem of causality and time series analysis for a number of years, decades, really, and they came out with this very important paper a couple of years ago, an alternative approach to assessing causality in complex systems. And this is the approach that I use to understand what's driving the long-term pattern and productivity in the system. So, briefly, I'm going to go over this. In dynamic systems theory, X and Y are causally coupled if they belong to the same dynamic system. In this case, the same attractor manifold. And the attractor manifold, an attractor could be a manifold or a point or a curve. It's just a system where physical properties tend to evolve towards, irrespective of initial conditions. Okay? And so MX and MY, I'll get to what these are in a second, these are what's called shadow manifolds, or these are constructions of original time series using lagged coordinates. And you build a library of shared points in time space. And that shouldn't have happened. Wrong button. Okay, so they borrow heavily from Taken's theorem, Taken's embedding theorem, which says you can develop a Lorenz type attractor just using lagged coordinates of a single time series. So here we have the original time series, X. Let's say this is the Palmer-Drought severity index. And then we have that minus 1 year, minus 2 years, 2 times lag. Okay? And so we can use these to actually recreate different time series with different lags. We can do this for, and this is the shadow manifold for X, and we could do the same thing for Y. And then see where they correspond if they share a common manifold. And then we can look for nearest neighbor points in the shadow manifolds of MX and MY. And the longer your time series, the denser the manifold becomes, and better your predictions of one time series versus the other. So, so in short, we're looking for the signature of time series Y in time series X. So in the case of climate versus above ground primary productivity, we're taking the long-term time series of, say, of Forb production, and then projecting it into the manifold of a climate variable and seeing if we can predict climate from it. And then and this is called cross-mapping. And they call, what they call convergence is, as you increase your time series length, your predictions should get better because it gets tighter. And here you have more opportunities for looking at nearest neighbors. And this is what they can call convergence. And here's an example where they took on one of the classic problems in fisheries and biology is what explains the patterns in long-term patterns in sardine and anchovy catches off the coast of California. And if you look at this, and if you have a background in ecology, it just strikes a classic competition. Well, obviously, the sardines are going up, and then anchovies get their chance. Well, they use convergent cross-mapping to test for those dynamics. And what they find is, so this is rho, this is correlation, and this is a time series length. This is not straight correlation. This is the strength of convergent cross-mapping predictions. There's no relationship between the two fish species. They don't interact. Rather, they're both forced by a common driving variable, in this case, which is sea surface temperature. As you can see, the predictions increase as you go through time. Okay. This is what I did for the time series here. So these are AMPP cross maps to climate. Here's total, forbs, and grasses. And the colors correspond to the different climate variables. You can see for total primary production in the system, the strongest predictor is the Palmer Drought Severity Index. And then there's some influences of winter. Here's late rain. Forbs have a particularly strong response, again, consistent with their stronger decline. And here's a similar response in grasses. The error bars represent uncertainty imposed by missing data in the time series. Okay, fair enough. But does this completely resolve it? There's other things going on. What about growing season length, for example? What about nitrogen deposition? What about CO2? Well, like I said, we have the opportunity to take advantage of this long-term snow experiment. Excuse me. And what this does effectively is it shortens the growing season length in an already very short growing season by 2 to 4 weeks. Significant. It insulates the snow. And it insulates the soils over a winter. It's a dramatic change, so it allows us to control for the length of the growing season in a way and things operating over the winter. Okay, so effects that could be happening for the winter and effects that are occurring during the winter but then sort of emerge later in the growing season. What do we find here? So here's just another shot. Here's early growing season. You can see there's still snow covering the ground in these. Here's lysimeters for measuring nutrient losses. This is work that Yuriko Yano's been doing, and that's a story for another time. We find precisely equivalent declines in long-term dynamics in these plots. And even stronger convergent cross-mapping with the Palmer Drought Severity Index and late rain in these two treatments. So here's ×4, and then here's ×2. By the way, these bands are just confidence. They're just showing uncertainty. So these are all the points that I put on the last time. These are all TABS plots over time. And so it's sort of a running mean. Okay. This helps us isolate late summer growing conditions as the culprit of the decline. What about nitrogen deposition? It's changed dramatically in many places in the world. Turns out, not so much around here. Here's the Yellowstone Tower. And if you take this point out, there's no significant change. And you can see there's an increase. These are very low numbers. These are background nitrogen deposition levels. And the same thing for nitrogen in the USGS database in snow. Haven't seen any consistent regional effects. Moreover, we have archived samples that go back over 20 years from the grassland, and so I've had these analyzed for nitrogen content and the 15N, 14N ratios of the tissues, which can be, in some cases, a very sensitive indicator of the change of in a nitrogen cycle. And again, we show no secular change in 15N or nitrogen. I don't think that nitrogen is really doing much yet in this system. How about CO2? So have these analyzed for 13C, heavy isotope of carbon. Now plants like to take up light carbon. And although imperfect, the delta 13C can be an approximation of water use efficiency in some cases. So what we see in grasses during drought or dry periods, as grass production goes down, they discriminate less, consistent with an increase in water use efficiency during these periods. We don't see this in forbs because these are mostly annual. We wouldn't expect that at all. And so this suggests that perhaps changes in water use efficiency as revealed by carbon isotopes have ameliorated some of the changes due to increasing aridity in the system. And if this hadn't occurred, if they hadn't dynamically responded like this, the crashes and biomass may have been even more severe. Okay. Well, what is the mechanism? The hypothesis is that we know that most production is occurring earlier in the growing season, but what late rain, as all the analysis I just showed, increasing aridity in the later season does, is it allows for an extension of photosynthesis, not necessarily biomass accumulation and growth, but allows them to stay green. That photosynthate can then be used to put below ground at the end of the season and kickstart the growth the next year. Over time, as you cumulatively reduce the amount of carbon going below ground, you should have cascading effects on future productivity. This is a hypothesis that we need to continue working on, but I think it's right now the most sound one for what's explaining these patterns. Okay. So what have we learned here? I think one of the most important ones is taking advantage of existing long-term data and observing how systems have responded to real and observed climate change and climate variability over the last half century, the period of major anthropogenic forcing in the climate system. And I think it suggests that we can consider some alternative techniques for assessing causality in these complex systems as well. Okay. Yep. So in this case, we find that increasing aridity has outpaced other change factors to modify the long-term pattern in productivity. Nitrogen deposition can't keep up, CO2 can't keep up. It's just getting drier, and the system has lost its capacity to take up carbon by half. Not true everywhere, but seems to be the case here. Okay, let's move from grasslands to forests. And I'm going to briefly introduce some work we just started in the Swan Valley that I'm really excited about. And so getting to this question, how do level patterns of nutrient limitation and primary production emerge from local organism-specific interactions, and how do these change as the ecosystems age? Okay, this is a root. And this diagram is from a theoretical paper by Roos, and she's done some of the most brilliant work on modeling the root-soil interface, looking at the physics of solute transport. So we have convection diffusion equations, and we basically model how roots interface with particles that are diffusing around them. And this has been a major way that we understand the nutrient uptake kinetics of roots. And this is something that, as of now, has not been incorporated in a good way in our system models or ecosystem models. The alternative approach is a phenomenological sort of approach where we do a top-down. We solve basically via mass balance what plant uptake must be to produce the emergent patterns that we see in the ecosystem. Okay, but these haven't been united, these two approaches, which I refer to as the micro and the macroscopic views of how plants take up nutrients from soil. So my colleague Stefan Gerber and I recently accomplished this. So we started with the solute transport framework of Roos and others. This is usually referred to as the Nye-Tinker approach, or Tinker and Nye approach. And so we have, diffusion contents and water-filled pore space and stuff you find in all these models, but lacking in most of these are actually a link to ecosystem-level processes of microbial mineralization of nutrients, losses, how water interacts with roots. Why is it that water just doesn't steal stuff from organisms more often? And so we are asking, under nutrient-limited conditions, how is it that plants are able to maintain such a tight nutrient cycle? And so the amounts of nitrogen, for example, that you see in stream waters is so sparingly small. And so our model, like others, just shows the profound effect of root diameter, and these are normalized concentrations in units of years in time. And so we can also assess the effects of mycorrhizae The long and the short of it, though, so roots draw down nutrients in a more effective manner when they're smaller, when they have small diameters. And this also changes, and so this makes sense in terms of a cylinder, which we model a system as. So we populate the soil and an ecosystem, a whole landscape with these cylinders. A simplification, of course, but addressing the huge diversity of root types and morphology is another challenge. Okay, so then we link this to ecosystem level patterns. So here's our solution. We analytically solved for root uptake, so which we call KP, so uptake rate constant of plants. And the 3/2, all this stuff derives from doing this in cylindrical coordinates. So this is the geometry of root uptake. So for the first time, we have an analytical solution for root uptake constant that can be put into more complex ecosystem models. And all it requires is knowing what the mass of the roots are, what the specific root length is, what the radius is, and what the depth of the soil is. And according to this, you should be able to solve what root uptake is. You can also do this from the top-down approach using ecosystem models like we've done, where we have our KP here, and we basically solve it like for mass balance. And they correspond pretty well. We're really excited about this. So we evaluated it using data collected from the long-term ecological research stations in the United States. We get pretty darn good correspondence. The different symbols are with nitrogen deposition or without nitrogen deposition. Doesn't seem to matter very much, at least at these sites. But so here we go. We have a theoretically sound mechanistic framework for defining nutrient root uptake. What do we do with that? And what is missing from this? One thing that's missing is we don't address the size of the plants or really the conditions in which they're living. Our solution is for the nutrient-limited case, so the steep part of the Michaelis-Menten curve, if you will. Well, how does this change as trees get older and bigger, and the ecosystems that they live in age, and the feedbacks they're in? Trees range from this and much smaller to this size, okay? And this, of course, is the study of metabolic scaling and allometry. And so here's just one example among many from a conversation between Inkvist and Reich, basically arguing about the role of nitrogen content in explaining the scaling between plant mass and some metabolic function, in this case carbon uptake, or could be respiration. So many of these patterns scale with a 3/4 power law. And this goes back to West and Galuli and others in their brilliant papers of the 90s that basically solved the problem up to then and assigned the patterns to basically how critical resources are passed through fractal networks in animals and plants. And this explains the branching patterns in our bodies and aortas and plants. And this has gone through a lot. Now people are trying to scale up to ecosystems. And we're going to take a stab at this too. But what they haven't linked to so far is basically how ecosystems respond to disturbance and how all these properties, our nutrient uptake solutions, change when you fundamentally disrupt the system. And this is a fundamental problem in modern Earth system models as well, in terms of really addressing the assumptions of assigning the 1850 pre-industrial state. Okay, so you have to assign initial conditions. Some people choose to let their pool sizes free because there's tremendous assumptions about this. And at all scales, addressing disturbance, which has been a cornerstone of ecological thought for half a century, really isn't in a lot of these models very well. nor is root uptake, or how these things change with the size of organisms or the age of the ecosystem. Okay, so what we want to do is link our model and evaluate our model in the context of a whole ecosystem, here as illustrated by soil organic matter and the tree, and how this changes over the course of time as trees and ecosystems get larger. Okay. This work is going to be primarily the work of my graduate student, Erin Klingborg. And so let's move up to the study site where we're going to do some of this. And I'd like to introduce the Swan Chronos sequence. And this is a system that is really made possible by the work of my colleague Travis Bloat at the Wilderness Society. So he has developed this database here of all the parcels in the Swan. And at each one of these points, we know the exact cutting history and silvicultural treatment. Most of the valley was cut in 1918, and in the last several, and then was owned by Plum Creek on about a 70-year rotation cycle throughout this whole area, and then has recently been turned over to the Forest Service, almost completely. And so we're addressing the northern end of the valley, which was cut first, and the southern end is more complex. We want to move down there, but just the two of us right now, so it's a lot of work, and they're doing a lot of prescribed burning down here. I mean, it's really cool, beautiful systems where they're burning into giant larch and ponderosa and everything, but we're focusing on the wetter end, where we are pretty sure about the disturbance history. So our sites range from the youngest sites that we could find, which are 14-year-old clear cuts, all the way up to 350-year-old full growth that has never been cut. We have tried to the best extent possible to control for complicating state factors. And so this is very preliminary, and this is mostly just sort of an introduction to the sort of things we're interested in. Don't have any conclusions yet on this. These are data from this summer, in fact. And so here is a classic allometric relationship between stem diameter and tree height. This is commonly used by foresters and theoreticians both to understand how forests are working. So in the color coding, we have very young forests, mature forests, which were cut in 1918, and then our old growth. And these trees are distributed across a number of different species. So what's wonderful about these sort of plots is you can see succession in action in these two numbers, the size of two allometric numbers about the size of trees. You can see a system's getting older. See a plicata, western red cedar moving in, it's a little sprinkled in through here, grand firs throughout, and then these big guys, Lyrax occidentalis, here, that probably derived from the last historical fires, very fire resistant, and these guys are the tallest guys on the block. So we have focused on a subset of these for intensive work, and we're doing a whole variety of things on these plant soil systems. And we want to link this to ecosystem level patterns of nutrient cycling and limitation in the system. Okay. So here is vegetation, biomass, in megagrams per hectare. This is calculated using species-specific allometric equations for across the sites. Here's litter biomass, how this is changing dynamically through time. And then here's the O horizon. And this is sort of a classic response that many people have observed. It's often referred to as Covington's curve, derived from work in New Hampshire in the 1970s. And this is often attributed to massive increases in respiration following disturbance. We actually still don't really know why this happens. It's a bit more complex. So that's basically what we have right now from the Swan. And we're really excited about the opportunity to link time, individual organism interactions, and ecosystem level emergent patterns and nutrient limitation at the site. And this is what we want to do. No big deal. Link root uptake organism scale allometry and scaling to ecosystem level patterns of change over time. This is the change in biomass over time, borrowed from my colleague Duncan Mingy. Here's detritus over time. Here's plant available nutrients over time. We will have all the data we need to link these together. So this will be a joint theoretical and empirical effort, and hopefully a feedback between the two. And I hope to present on what we come up with in the next couple of years. So to recap, basically how changes to background aridity and other factors. In this system, the Bangtail grassland system, it appears that changes in background aridity are really having a big effect. It has really severely depressed the ability for these systems to take up carbon over time, and other factors have not ameliorated this long-term change. And it really, to me, emphasizes the importance of long-term data monitoring, which is becoming less and less funded. And the second, I think we are just now developing sort of the tools and framework to evaluate some very interesting interactions and coupling between how organisms work and interactions and feedback with the ecosystems in which they live. It's very preliminary. And that's what I have. So, thank you.
Lots of time for questions.
Yeah.
So thank you for that talk. I'm glad to see the work coming from the Bangtails. So towards the end, you had a picture of a LI6400.
Yeah.
Kind of perked my interest a little bit. I was thinking of ways you could possibly look at the mechanism that you're describing, ways to look at if they're putting extra carbon below ground. And I was thinking that if you went out with the LICOR, it'd be really labor intensive. You'd probably not get a really good representation because it takes so long. But one thing you could potentially do is look at sugars and starches. So in the middle of the growing season, if the plant is photosynthesizing, it's probably using a lot of the sugars and starches for growth. But if it's photosynthesizing more after the rains come and it's putting it more below ground, you'll see more higher concentrations of sugars and starches being stored. And so you could go out, collect a lot of samples over time and over space, and just look at non-structural carbohydrates as some indication of whether or not it's storing it. And you could potentially pick it up the next spring when the plants are coming back up again.
That's a great idea. Can we do that relatively non-destructively?
No, you have to clip a leaf, but you don't need very much material.
Yeah. Every little square centimeter is precious.
Yeah, you could get away with 100 grams of dried leaf material.
Let's do it.
Okay.
Andy.
Yeah, I do echo John's statement. It's really great to see the analysis of that data set that Ted got going. It's really fascinating. So really great work.
Thank you.
Just a context sort of comment maybe. When we use PRISM data to look at an aridity index for the last 110 years for GYE as a whole, it's really that it actually, aridity basically decreased during the first 60 to 70 years of that. And instead of inflection, it started to increase.
Mm-hmm.
That suggests, that and other climate data, suggest decadal scale oscillation as being typical in the system, and then possibly, a human-induced signature now starting to emerge. I guess the point is, if in fact the data that you represented was typical of the system as a whole and representative of a 50% decline in biomass production, over historic times, that would be quite shocking from a ungulate perspective, for example. On the other hand, if this is mostly reflecting decadal scale oscillation, then it's well within some sort of range of natural variability. I just wonder. I think that what you've done is so cool. It'd be quite nice to now begin to try to think about it in a longer-term context, to be able to try to separate the natural decadal oscillation in the system signature from a human-induced climate change signature.
Yeah.
My guess is, I mean, just based on what we know about aridity, I don't think that elk have half as much forage as they have for the average of the last 200 years. But rather, Tad's time period pretty much captures the highest productivity period and the lowest in the last 300 years. I mean, so those are just alternative hypotheses to test.
I think it's a really important number of points you've raised, and I don't know if they're quite alternative hypotheses. I think you're absolutely right. What we may be seeing is just sort of one part of a phase of a much longer term sort of system. And I know I've looked at the longer term PDSI records as well, and basically that downtrend occurs right at the beginning of our record. And that's what I'm focusing on, just because that's where we have productivity data from. But indeed, we could be seeing just a normal part of a longer-term decadal century type oscillation in climate and coupling with production, and it's still very difficult to disentangle that from the most recent anthropogenic forcing. Yeah.
Yeah, great stuff. I was curious about the increased density of herbivory, ungulates, just from changes in hunting practices. Would that change or did you take that into account, or would it affect your results? Just deer density in the area.
I rely on Tad for the observations of the animals over the last 4 decades, and the site was grazed historically, like much of this area, but hasn't been grazed since the 1930s. So I feel pretty confident that the pattern we're seeing is not a release from livestock grazing. The area is fully accessible by the full range of large animals that exist there. The area I don't think was really ever historically grazed by bison, but elk and deer have full access to the site. There's also a pocket gopher thing going on up there that's sort of complex, especially under the snow. But I don't know the answer to that question. I think it's very interesting, though. Tony.
I love the aridity setup here, and I'm curious, because every square centimeter is so valuable, do you have ideas for how to basically image what's going on below ground in as non-destructive a way as possible?
Yeah. The first way I want to do it, I'm working on right now, is developing simple models to make sure that it makes sense from a carbon balance perspective as a legitimate, as a parsimonious hypothesis. In terms of going in and measuring, I think Jaw's suggestion sounds great. I mean, I guess you could go in with mini risotrons, other instruments that I've never used, and check it out from there. Yeah, we really don't know, but my sense is that that overall, so one question that could be asked is, well, maybe they're just shunting all their carbon below ground earlier, and in fact, the total carbon hasn't changed very much. I really think that's unlikely. I think we're seeing a cumulative effect of a decrease of ANPP over time. And essentially, the above ground biomass is what is returned to soil every year. And we do have archived samples of soils that I still intend to run for carbon density and C13, and those have not been analyzed yet, but it's not high resolution. It's like a few times over the last 25 years. But yeah, I would love to hear ideas on how we could do that.
I know you kind of championed the idea that the bangtail would be a reserve or a LTER or a macro system site, and how well protected is it, and what's happening with those ideas?
Yeah.
Who owns it, for one thing?
Forest Service.
It's Forest Service.
It's a special interest area, and it is protected. At least our core site is protected from pretty much everything. We maintain the fences so that cattle don't get in there, et cetera. But yet it's they designated it as a special interest area on the basis of unique fossils, Native American. So there's some cool, what are they, tarsiers in the rocks around there, and Native American artifacts, and then the long-term work of TADS and IBP. I have tried to get an LTREB going there over the last several years, and it has been unsuccessful, and I don't really know why. So yeah, I had a reviewer say this is a very unusual system for an LTREB, but didn't say why. So I think it's a pretty good system for a long-term research and environmental biology site, which is sort of a mini LTER. And I'm going to try again. And I'll keep trying because I think it's a valuable system, to keep the monitoring going and developing new measures to try to understand what's going on. Well, thank you.
Well, great. Thanks, Jack, very much. That was great.